Welcome to our deep dive into the fascinating world of DNA, the molecule that holds the blueprint of life. My name is Sabina, and I am thrilled, absolutely thrilled, to be your guide today as we unravel the secrets of this incredible structure. Think about it. Every living thing, from the tiniest, most minuscule bacterium to the largest, most colossal blue whale, and yes, even you and me, relies on this amazing molecule called deoxyribonucleic acid, or DNA for short. It's the instruction manual, the master plan, the very essence of what makes us us. It dictates our eye color, our height, and so much more. It's truly the architect of life. Today, we're going to journey into the microscopic realm, a place smaller than you can even imagine, to understand what DNA is, how it's built with such precision, what it does with all its incredible information, where it resides within our cells, and how it makes perfect copies of itself. It's like we're embarking on a molecular safari, and trust me, the sights you're about to see are astonishing, mind-boggling even. We'll see how these tiny threads, packed with an immense amount of data, orchestrate the entire symphony of life, ensuring everything runs smoothly. It's a story of chemistry, biology, and awe-inspiring complexity, all elegantly packaged within almost every cell of your body. So get your thinking caps on, because we're about to explore the fundamental molecule that connects all life on Earth. We'll break it down piece by piece, concept by concept, so that by the end of this, you'll not only understand DNA, but I hope you'll also share my profound sense of awe for its sheer elegance and its undeniable power. It's going to be quite an adventure, an exploration into the very core of existence. So let's get started. Science is all around us, it's in us, and understanding DNA is absolutely key to understanding ourselves and the wonderfully complex world we live in. Consider this your personal invitation to the amazing world of genetics. All right, let's dive right into the architecture of DNA. When scientists first figured out what DNA looked like, it was a groundbreaking moment. Picture this. DNA is a double helix. Now, what does that mean? It means it has two long strands, not just floating around. Oh no, these strands are twisted around each other, kind of like a spiral staircase, or even better, a twisted ladder. This twisting shape is super important. It's not just for show. It helps make the DNA molecule stable and compact, allowing a huge amount of information to be stored in a tiny space. Isn't that amazing? A ladder, but make it spiral. Now let's talk more about this twisted ladder analogy, because it's a really good one. Think of a regular ladder. It has two long side supports, right? And then it has rungs that go across, connecting those sides. Well, DNA is very similar. The two long twisting strands form the sides of our molecular ladder. And then there are rungs that connect these two strands. We'll get to what those rungs are made of in just a moment, but for now, just keep that image in your mind. A long, elegantly twisted ladder holding the secrets to life itself. This structure was famously figured out by Watson and Crick with crucial data from Rosalind Franklin. Science! So we have these two strands coiling around each other in a beautiful dance. What are these strands actually made of? Well, each individual strand is a polymer. That's a science word for a long chain made up of many, many repeating smaller units, all linked together. Think of it like a very, very long train, where each carriage is one of these repeating units. Or perhaps a pearl necklace, where each pearl is a building block. These repeating units in DNA have a special name. They're called nucleotides. Say it with me. Nucleotides. They are the fundamental building blocks of these DNA strands. Each one of these nucleotides, these essential building blocks, is itself made of three distinct parts. It's like a little three-piece construction kit. First, there's a sugar molecule. Not the kind of sugar you put in your cereal, though. This is a specific type of sugar called deoxyribose. That deoxyribo part is actually where DNA gets the D in its name, deoxyribonucleic acid. This sugar forms a key part of the backbone, or the sides of our twisted ladder. It's a 5-carbon sugar, and its structure is crucial for the overall shape and function of the DNA molecule. The second part of our nucleotide construction kit is something called a phosphate group. A phosphate group is made of phosphorus and oxygen atoms. Now, these phosphate groups are really important because they link the sugar of one nucleotide to the sugar of the next nucleotide in the chain. This sugar-phosphate connection creates that long, strong backbone for each DNA strand. So if the sugars are like the vertical posts of our ladder sides, the phosphates are the connectors holding those posts together in a long, continuous line. This backbone is incredibly strong, protecting the precious information inside. And the third and perhaps most exciting part of each nucleotide is called a nitrogenous base. Nitrogenous just means it contains nitrogen atoms. These bases are the rungs of our DNA ladder we mentioned earlier. There are 4 different types of these nitrogenous bases in DNA, and they are often represented by letters: adenine, which we call A, thymine, which we call T, cytosine, which we call C, and guanine, which we call G, A, T, C, and G. These 4 letters form the alphabet of the genetic code. It's this sequence of bases that carries all the instructions for building and operating an organism. Let's look a little closer at these amazing nitrogenous bases. Adenine, A, and guanine, G, belong to a class of chemicals called purines. Purines have a double ring structure, making them a bit larger. Think of them as the slightly bulkier letters in our genetic alphabet. Their specific chemical makeup is what allows them to interact in very particular ways with the other bases, which we'll explore very soon when we talk about base pairing. It's all about the shape and the available chemical bonds, you see. Molecular structure dictates function. Then we have cytosine, C, and thymine, T. These 2 bases belong to a different class of chemicals called pyrimidines. Unlike the purines, pyrimidines have a single ring structure, so they're a bit smaller, a bit more streamlined, if you will. Again, their unique shapes are absolutely critical. The subtle differences in these molecular structures are what make the precise pairing between bases possible, forming the rungs of our DNA ladder and ensuring the genetic code is read correctly. It's like having perfectly fitting puzzle pieces. So, to recap this amazing structure, each nucleotide has a sugar, deoxyribose, a phosphate group, and one of the 4 nitrogenous bases, A, T, C, or G. The sugar and phosphate groups link together to form the long backbone of a DNA strand, the sides of our ladder, and projecting inwards from this backbone from each sugar is one of those nitrogenous bases. These bases are ready to pair up with bases from the other strand, forming the rungs. It's an incredibly elegant and efficient design, perfected over billions of years of evolution. Imagine millions, even billions of these nucleotides all linked up in a precise sequence, forming one strand and then another strand, perfectly complementary, pairing up with the first one. And then the whole thing twists into that iconic double helix shape. That, my friends, is the fundamental structure of DNA. It's strong, it's stable, it stores vast amounts of information, and it's the starting point for understanding everything else about how life works. Consider the sheer amount of information packed into such a tiny structure. It's mind-blowing. Now that we understand the magnificent structure of DNA, with its 2 strands forming a twisted ladder, let's zoom in on those rungs. The rungs of our DNA ladder are formed by pairs of nitrogenous bases, and this pairing isn't random at all. Oh no, it's incredibly specific, like a secret handshake that only certain partners know. This specificity is absolutely crucial for DNA to do its job correctly. If the pairing was haphazard, the genetic code would be a jumbled mess. Think about it. Precision is key when you're dealing with the blueprint of life itself. The rule is beautifully simple, yet profoundly important. Adenine, A, always, and I mean always, pairs with thymine, T, and cytosine, C, always pairs with guanine, G. So A goes with T, and C goes with G. It's like they're best friends who only hang out with each other. An A on one strand will always find a T on the opposite strand to link up with. Similarly, a C on one strand will always seek out a G on the other. This is known as complementary base pairing, and it's a cornerstone of DNA's structure and function. Why this specific pairing? It's all down to the chemical structures of the bases themselves, their shapes, and the way they can form connections. Remember how we talked about purines, A and G, with their double ring structure, and pyrimidines, C and T, with their single ring structure? Well, a purine always pairs with a pyrimidine. This ensures that the width of each rung on our DNA ladder is consistent. If 2 purines tried to pair, they'd be too bulky. If 2 pyrimidines tried to pair, they'd be too narrow, creating a wobbly ladder. Nature's design is so clever. These base pairs, AT and CG, are held together by special kinds of chemical bonds called hydrogen bonds. Now, hydrogen bonds aren't the strongest bonds out there, like the covalent bonds holding the atoms within a nucleotide or the sugar-phosphate backbone together. Individually, a hydrogen bond is relatively weak. But, and this is a big but, in a DNA molecule, there are millions, even billions of these hydrogen bonds holding the two strands together. It's like a zipper. Each individual tooth of the zipper isn't super strong, but altogether they hold things very securely. Let's look at the specifics of these hydrogen bonds. The pair between adenine A and thymine T is held together by two hydrogen bonds, just two. It's a good solid connection. Now, the pair between cytosine C and guanine G is a little bit different. The CG pair is held together by three hydrogen bonds. Three! This means the connection between C and G is slightly stronger than the connection between A and T. This difference in bond strength can be important in certain biological processes affecting how easily regions of DNA can be unzipped. The beauty of these hydrogen bonds is that while they are strong enough collectively to keep the two DNA strands together and maintain the double helix structure, they are also weak enough individually to be unzipped or separated when needed. And when does DNA need to be unzipped? Well, for important processes like DNA replication, making a copy of itself, or transcription, reading the genetic instructions to make proteins. The cell has special machinery, enzymes, that can come along and temporarily break these hydrogen bonds, allowing the strands to separate. This precise AT and CG pairing rule means that the two strands of a DNA molecule are complementary to each other. If you know the sequence of bases on one strand, you automatically know the sequence of bases on the other strand. For example, if one strand has the sequence AATGCCG, then the complementary strand must have the sequence TTACGGC. Isn't that neat? It's like having one half of a code and being able to perfectly deduce the other half. This complementarity is fundamental to DNA's ability to replicate and repair itself accurately. Consider the implications of this complementarity. It's like having a backup copy built right into the structure. If one strand gets damaged, the cell can often use the other strand as a template to repair the damage accurately. This ensures the integrity of the genetic information. The sequence of these base pairs, these A's, T's, C's and G's strung out along the length of the DNA molecule is what constitutes the genetic code. It's a four-letter alphabet that spells out the instructions for building and maintaining an entire organism. Millions of these letters in a row. The discovery of this base pairing rule was a monumental breakthrough. It immediately suggested how DNA could carry information and how it could be copied. James Watson and Francis Crick, when they proposed their double helix model, recognized the significance of this pairing. It was a key piece of the puzzle that unlocked our understanding of heredity and genetics at the molecular level. Suddenly, the abstract concept of a gene had a physical chemical basis. It was revolutionary, to say the least. So, to sum up base pairing, adenine pairs with thymine, AT, using two hydrogen bonds, and cytosine pairs with guanine, CG, using three hydrogen bonds. This specific pairing ensures the strands are complementary, that the ladder rungs are a consistent width, and that the genetic information is stored securely yet accessibly. It's an elegant system, a marvel of molecular engineering that allows life to perpetuate itself with remarkable fidelity. The dance of these bases is the dance of life itself. Chapter 3. The Master Plan, DNA's Crucial Functions. Now that we've explored the beautiful structure of DNA and the precise rules of base pairing, let's get to the really big question. What does DNA actually? Why is it so incredibly important? Well, hold on to your hats, because DNA has some of the most crucial jobs in the entire biological world. Its primary function is to carry the instructions, the genetic information needed to build and operate a living organism. It's the master blueprint, the ultimate instruction manual for life. One of the most fundamental things DNA contains is genes. You've probably heard of genes before. A gene is a specific segment of DNA, a particular sequence of those A's, T's, C's and G's that typically holds the instructions for making a specific protein. Proteins are the workhorse molecules of the cell. They do almost everything. They form structures, like your hair and muscles. They act as enzymes to speed up chemical reactions. They carry signals. They transport molecules and so much more. So genes in DNA tell the cell what proteins to make. Think of DNA as a giant cookbook, and each gene is a single recipe in that cookbook. each recipe provides the exact instructions for making one particular dish, in this case, one particular protein. The sequence of bases in a gene determines the sequence of amino acids that will be joined together to make that protein. And the sequence of amino acids determines the protein's structure and ultimately its function. It's an incredibly precise system, ensuring the right proteins are made to do the right jobs. Beyond just telling the cell how to make proteins, DNA is also responsible for heredity. This is a huge one. Heredity is the passing of traits from parents to their offspring. Why do you look a bit like your parents or share certain characteristics with your siblings? It's because you inherited your DNA from them. You get half of your DNA from your biological mother and half from your biological father. This DNA carries the genes that determine many of your traits, from physical characteristics like eye color and height to predispositions for certain health conditions. This process of passing traits from one generation to the next is fundamental to life and evolution. The faithful copying of DNA, which we'll talk about in detail later, ensures that offspring receive a complete set of genetic instructions from their parents. Sometimes small changes or mutations can occur in the DNA sequence, and these can lead to new traits. Over vast timescales, this process of inheritance, combined with mutation and natural selection, drives the evolution of species. So DNA isn't just about individuals, it's about the continuity and diversification of life itself. Another critical function of DNA is to guide the cell in producing the correct proteins, not just in the right way, but also at the right times and in the right amounts. A cell doesn't need to make all of its proteins all the time. For example, a skin cell needs different proteins than a brain cell, and a cell might need certain proteins only during specific stages of development or in response to particular environmental signals. DNA contains regulatory sequences that control when and how much a gene is turned on or turned off. This is called gene regulation, and it's incredibly complex and vital for normal development and function. Imagine our cookbook analogy again. Not only does it have recipes, but it also has notes about when to cook certain dishes, for which occasions, and how many servings to prepare. DNA, through its regulatory regions, manages this intricate cellular kitchen with remarkable precision. This ensures that cells can specialize, respond to their environment, and maintain a stable internal state, a concept known as homeostasis. Without this precise control, cells and entire organisms would descend into chaos. So, DNA is not just a static storage molecule. It's an active participant in the life of the cell, constantly being read and interpreted. The information flows from DNA to another molecule called RNA through a process called transcription, and then from RNA to proteins through a process called translation. This is often referred to as the central dogma of molecular biology. DNA makes RNA, and RNA makes protein. It's the fundamental pathway for how genetic information is expressed and used to build and operate a living system. The sheer amount of information stored in DNA is mind-boggling. The human genome, for instance, which is the complete set of DNA in a human, contains about 3 billion base pairs. If you were to type out all those A's, T's, C's, and G's, it would fill thousands of books. And all of that information is packed into the nucleus of almost every tiny cell in your body. It directs your growth from a single fertilized egg into a complex multicellular organism, and it keeps you running every second of every day. Ultimately, the function of DNA is to ensure the continuity of life, the faithful transmission of biological information across generations, and the precise orchestration of cellular activities. It's the molecule that defines what an organism is and how it functions. From the simplest virus, some of which use RNA, but the principle of genetic material is similar to the most complex animal, DNA, or a similar nucleic acid, is at the heart of it all. It's the common thread that links all known life, a testament to a shared evolutionary history. Science is all about understanding these fundamental principles. Chapter 4: DNA's address. Where does it live? We've talked about what DNA is and what it does, but where exactly do we find this all-important molecule within a cell? Does it just float around anywhere? Not usually. The location of DNA is very specific, and it actually differs depending on the type of cell we're looking at. The two main categories of cells are eukaryotic cells and prokaryotic cells, and they house their DNA in distinct ways. Let's explore DNA's cellular address. First, let's consider eukaryotic cells. These are the types of cells that make up more complex organisms like plants, animals, fungi, and protists, including us humans. In eukaryotic cells, the vast majority of the DNA is neatly packaged and protected inside a specialized membrane-bound compartment called the nucleus. The nucleus is often called the control center or the brain of the cell, and for good reason. It houses the cell's precious genetic library, the DNA. This separation of DNA within the nucleus provides an extra layer of protection and organization. Inside the nucleus of a eukaryotic cell, the DNA isn't just a loose tangle of strands. Oh no, that would be far too messy and prone to damage. Instead, the long DNA molecules are highly organized and compacted. They are wrapped around proteins called histones, much like thread wrapped around a spool. This DNA protein complex is called chromatin. When the cell is preparing to divide, this chromatin condenses even further to form visible structures called chromosomes. You might have seen pictures of these X-shaped chromosomes. That's supercondensed DNA. This compartmentalization within the nucleus is a hallmark of eukaryotic cells. It allows for more complex regulation of gene expression because the processes of reading DNA, transcription, and making proteins from that information, translation, are separated in space and time. Transcription happens inside the nucleus where the DNA is. Then the RNA message travels out of the nucleus to the cytoplasm, where the ribosomes, the protein-making machinery, are located and translation occurs. This separation adds layers of control. Now, not DNA in a eukaryotic cell is in the nucleus. There's a tiny bit more. Eukaryotic cells also have small energy-producing organelles called mitochondria. You might know them as the powerhouses of the cell. Plants and algae also have chloroplasts, which are responsible for photosynthesis. Amazingly, both mitochondria and chloroplasts have their own small circular DNA molecules. This mitochondrial DNA, mtDNA, and chloroplast DNA, cpDNA, carry genes specifically for the functions of these organelles. It's like they have their own little instruction manuals. This is fascinating evidence for the endosymbiotic theory, which suggests these organelles were once free-living bacteria. What about the other main type of cell, the prokaryotic cell? Prokaryotes are simpler, generally smaller cells, and they include bacteria and archaea. Unlike eukaryotes, prokaryotic cells do have a nucleus. There's no separate membrane-bound compartment for their DNA. So where is their DNA then? In prokaryotes, the DNA is found in a region of the cytoplasm, the main body of the cell, called the nucleoid. Nucleoid means nucleus-like, but it's important to remember it's not a true nucleus because it's not enclosed by a membrane. The main DNA molecule in most prokaryotes is typically a single, large, circular chromosome. Imagine a long DNA double helix that has its ends joined together to form a continuous loop. This circular chromosome is still highly organized and compacted, even without a nucleus, often supercoiled and associated with some proteins to help it fit inside the relatively small cell. Even though it's in the cytoplasm, it's generally confined to this nucleoid region because there's no nucleus to separate the DNA from the rest of the cellular machinery. The processes of transcription, reading DNA into RNA, and translation, making proteins from RNA, can happen almost simultaneously in prokaryotes. As the RNA message is being made from the DNA, ribosomes can latch onto it and start making proteins right away. This allows prokaryotes to respond very quickly to changes in their environment, which is a big advantage for these often fast-growing organisms. Besides their main circular chromosome, many prokaryotes also have small circular pieces of DNA called plasmids. Plasmids are much smaller than the main chromosome and can carry a few extra genes. These genes often provide some sort of advantage, like antibiotic resistance or the ability to metabolize unusual nutrients. Plasmids can replicate independently of the main chromosome and can even be transferred between bacteria, which is a way they share genetic information. It's like trading useful software updates. So, to summarize DNA's location in eukaryotic cells like ours, the majority of DNA is linear, organized into multiple chromosomes, and housed securely within the membrane-bound nucleus, with a little extra in mitochondria and chloroplasts. In prokaryotic cells, like bacteria, the main DNA is typically a single circular chromosome located in the nucleoid region of the cytoplasm, and they may also have plasmids. Understanding where DNA resides helps us understand how cells are organized and how they manage their genetic information. It's all about location, 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 even at the cellular level. Chapter 5. Making copies, the marvel of DNA replication. One of the most astounding properties of DNA, something truly at the heart of life itself, is its ability to make exact copies of itself. This process is called DNA replication. Think about it. Every time a cell divides to make two new cells, whether it's for growth, repair, or reproduction, each of those new cells needs a complete and accurate set of DNA instructions. If the DNA wasn't copied correctly, the new cells wouldn't function properly and life as we know it couldn't exist. So, DNA replication is absolutely fundamental. The magic of DNA replication lies in its very structure, particularly the complementary base pairing we talked about. A with T and C with G. Because the two strands are complementary, each strand can serve as a template or a pattern for building a new partner strand. This is the genius of the design. When Watson and Crick first proposed the double helix structure, they immediately saw how this complementarity could explain replication. It was a eureka moment for biology. So how does this copying process actually happen? It's a highly orchestrated molecular dance involving a whole team of specialized enzymes and proteins. The first step is that the DNA double helix needs to unwind and unzip. An enzyme called helicase, think of it like a molecular zipper opener, moves along the DNA, breaking those hydrogen bonds between the base pairs, causing the two strands to separate, creating what's called a replication fork, a Y-shaped region where the new strands will be synthesized. Once the strands are separated, each original strand acts as a template for the synthesis of a new complementary strand. Another amazing enzyme, the star of the show really, called DNA polymerase, gets to work. DNA polymerase reads the sequence of bases on the original template strand and then adds the correct complementary nucleotides to build the new strand. For example, if it sees an A on the template strand, it will add a T to the new strand. If it sees a G, it will add a C, and so on, following those strict base pairing rules. DNA polymerase is incredibly accurate, like a highly skilled scribe, but it's not absolutely perfect. It makes a mistake only about once every billion or so nucleotides it adds. That's pretty amazing precision, and even when it does make a mistake, DNA has proofreading mechanisms. DNA polymerase itself can often detect and correct its own errors. Other repair enzymes are also constantly scanning the DNA for damage or mismatches and fixing them. This ensures the fidelity of the genetic information is maintained to an astonishing degree. Now, there's a little quirk in how DNA polymerase works. It can only add new nucleotides in one specific direction along the DNA strand, from the 5' end to the 3' end for you chemistry buffs. Because the two strands of the DNA double helix run in opposite directions, they are anti-parallel. This means that one new strand, called the leading strand, can be synthesized continuously, smoothly following the unzipping replication fork. It's like a straightforward highway drive. However, the other new strand, called the lagging strand, is synthesized in a more complicated way, because it has to be made in the wrong direction relative to the movement of the replication fork. It's synthesized discontinuously in short fragments called Okazaki fragments, named after the scientists who discovered them. Each fragment is made, and then another enzyme, DNA ligase, think of it as molecular glue, comes along and joins these fragments together to create a complete continuous strand. It's more like building a road in sections. The result of this intricate process is that from one original DNA double helix, you get two identical DNA double helices. Each new DNA molecule consists of one original parent strand and one newly synthesized daughter strand. This mode of replication is called semi-conservative replication, because each new molecule conserves half of the original molecule. This was another brilliant prediction confirmed by experiments, showing just how elegant the system is. This ability of DNA to replicate itself is absolutely essential for cell division. There are two main types of cell division, mitosis and meiosis. Mitosis is the process by which most of our body cells divide for growth and repair. Before a cell undergoes mitosis, its DNA replicates so that each of the two resulting daughter cells receives a full, identical copy of the genome. This ensures all your body cells, with a few exceptions, have the same genetic information. Meiosis, on the other hand, is a special type of cell division that produces gametes, sperm and egg cells, for sexual reproduction. In meiosis, DNA replication also occurs beforehand. But then there are 2 rounds of cell division, resulting in cells that have half the amount of DNA as the original cell. When a sperm and egg fuse during fertilization, the normal amount of DNA is restored in the new organism, which then inherits a unique combination of genes from both parents. DNA replication is the starting point for all of this incredible inherited diversity. Chapter 6, The Never-Ending Story of DNA, and there you have it. We've journeyed from the elegant double helix structure of DNA through its precise base pairing rules to its crucial functions as the blueprint of life, its specific locations within different cell types, and finally its amazing ability to replicate itself. It's a molecule of incredible complexity and beauty, the very foundation upon which all known life is built. From the simplest bacterium to the intricate workings of the human brain, DNA is the common thread, the ancient language that connects us all. It's truly awe-inspiring to think about the sheer amount of information and the precision engineering packed into these tiny molecular strands. Understanding DNA has revolutionized biology and medicine. It's allowed us to understand genetic diseases, develop new therapies, trace evolutionary histories, and even solve crimes. And the exciting part, we're still learning. The field of genetics and molecular biology is constantly evolving, with new discoveries being made all the time. There are still so many secrets hidden within our genomes, so many intricate mechanisms to uncover. The story of DNA is far from over. In many ways, we're just beginning to truly understand its full implications and potential. So I hope this exploration has sparked your curiosity and given you a deeper appreciation for this remarkable molecule. The world of science is full of wonders, and DNA is undoubtedly one of its most profound. Keep asking questions, keep exploring, and never lose that sense of amazement for the natural world. There's so much more to discover, both out there in the universe and right inside our very own cells. Thanks for joining us on this journey through the incredible world of DNA. Stay curious and keep exploring the wonders of science.